Hey, good people. I'm so excited today. We are going to look at urinification 101. When we study body structures, we look at how they are, we look at the structure and the function. And today we are going to look at the kidney, but we are not going to really look at the structural aspects of the kidney, but rather we are going to delve more into the function. But we have to appreciate that the kidney has a variety of roles, many of them. And today we are going to look at urinification. That's why the name of the video is Urinification 101. As we have said, that urinification takes place in the kidney. This is how the kidney looks like. But again, in biology, in physiology, we always have to appreciate the order or the levels of organization. That for us to understand the process, we need to understand the basic unit or the organ in which that process occurs. And in this case, it is a process taking place in the kidney. Therefore, we need to understand the basic structure of a kidney. And then the basic structure of a kidney is called a nephron. And here it is. Now, a nephron has parts. This is how it looks like, but let us try to understand what the parts of the nephron are. We have the glomerulus. This is called the glomerulus. Many of them are called glomeruli. And below it we have the Bowman's capsule. This is the Bowman's capsule. It's some kind of a U. Now we see that we have tubes going from here to here up to this side. So we can realize that these tubules are highly coiled. This is coiled. This is also coiled. We have some degree of coiling here. So this PCT is called proximal convoluted tubule. It's because it's in close proximity with the glomerulus. So hence it's called the PCT, proximal convoluted tubule. Then here we have the DCT, the distal convoluted tubule. It is far away from the what? From the glomerulus. So it is at the distal end. Now, things you have to understand about the glomerulus, it is supplied with blood, because what is happening during urinification, it's simply the filtration of blood. It happens here in the glomerulus, as we are going to see. So that means blood comes in the glomerulus and leaves the glomerulus. So what we need to understand is that we need to understand the blood supply to and from the glomerulus. So that brings us to two blood vessels. One that brings blood into the glomerulus, it is called the afferent arteriole. Then the one that takes away from the glomerulus is called the efferent arteriole. So, one thing, there are atomical differences between the afferent arteriole and the efferent arteriole. So one of these anatomical differences is that the afferent arteriole is larger than the efferent arteriole. So meaning that the blood vessel bringing blood into the glomerulus is wider than the blood vessel that takes away blood in the glomerulus. So the coming in is greater than the coming out. And that has a physiological advantage. So what that does is that it leads to a high pressure in the what? In the glomerulus. Isn't that cool? So now, going at the proximal convoluted tubule, PCT, it has carrier proteins, as we are going to see some processes occur there. Then we have the loop of Henle, which has two parts, as you can see. You have this part going down, that is called the descending limb of the loop of Henle. We also have this part going down, going up, it's called the ascending limb of the loop of Henle. This whole thing here is called the loop of Henle. So now, let us delve into urinification 101. Simply, in simplicity, urinification is the process by which urine is produced. You get? So, it involves three steps. There's ultrafiltration, there's selective reabsorption, and there's also tubular secretion. Tubular secretion. So now, what is ultrafiltration and where does it take place? Ultrafiltration takes place into the glomerulus. So we need to understand ultrafiltration. We all understand filtration, what it means is having a sieve, certain things pass through the pores and others don't pass through. So now, ultrafiltration happens. Ultrafiltration is simply filtration under high pressure. And it happens within the glomerulus. So now, how does filtration under high pressure happen in the glomerulus? So one thing we need to understand, remember I said the blood supply to the glomerulus is way too much than what goes away from the glomerulus. The blood coming in is way too much than the blood going out. So what that causes is, it causes high pressure in the what? In the glomerulus. So that blood pressure created, what it does is that it forces molecules out of the glomerulus into the Bowman's capsule. That's actually filtration. It's a filtration. High pressure created in the glomerulus results in two molecules leaking from the glomerulus into the Bowman's capsule. But it's not every molecule that passes through. What we need to understand in the anatomy of the glomerulus is that it has small pores, it has small holes inside there. So that means it is only small molecules that can pass through. And those small molecules include things like water, H2O, ions like sodium ions, potassium ions, and other mineral ions. We have sugars, amino acids, and things like urea. So things like proteins, which are large, or any other large, carbohydrates, antibodies, and any other large molecules are not filtered during ultrafiltration. If they do, it means you have a problem and you have to seek medical attention. So now we have seen that the high pressure created in the glomerulus, it simply means what gets out of the glomerulus is coming out of blood. This is blood being filtered. So the high pressure of blood in the glomerulus results into filtration of amino acids, sugars, small sugars, and mineral salts and water. But if blood is losing these things, doesn't blood need these things? Because let me give you an example. One of the things that are filtered out includes zinc ions. Doesn't the blood need zinc ions? Because zinc ions activate the enzyme carbonic anhydrase, which is involved in the transport of respiratory gases. You get? These molecules like sodium and potassium being filtered out are needed in transporting, in generating membrane potentials for certain physiological processes. So if they are being filtered out of blood, what are we going to do? Because I guess we need them. So now, what does the body do after filtering them out? Watch out for the next video.